So good morning, everybody. It's my pleasure to continue the session on late-stage clinical trials. And I think based on the titles of the topics, we will have some additional very exciting progress to be presented. And well, actually, it's already the third lecture of Dr. Desai, who will going to present on the peptide-based delivery for polypeptides that bypass the endosomal uptake pathway. So I'm very curious, and please take the floor.
So the original speaker of this talk, Dr. Gilles De Vita, is not here. Unfortunately, his mother is very ill, so he couldn't make it. So I'm going to replace him, but hopefully I can do justice to the science. So this is technology related to peptides as a platform for delivery, and these are specifically designed peptides. And for gene delivery, siRNA, plasmid DNA, using gene editing tools, etc. And this technology, I think you've heard quite a bit about this in the last several days, but delivery is a key issue. We've heard some successes with the siRNA delivery, some maybe limited success with gene delivery, and then moving on to cell therapies like CAR-T and then gene editing. There's still quite some limitations in terms of the type of technologies that are used. As you know, for non-viral vector technology, they use physical methods like electroporation, etc., for gene delivery into cells, which is not sort of quite optimal. So the technology I'm going to talk about is based in the company called AADi. And this is a new company, and it is based on the peptide-based delivery. And this technology was developed by Gilles De Vita, who was formerly at the CNRS in Montreal, in France. And then we take this technology from there and into the company AADi, and then we're going to be developing it for different applications and different indications. So the technology is based on the design of peptides that are amphipathic and that have specifically designed hydrophobic areas, electrostatic areas, aromatic areas that can serve as contacts to different cargoes that you want to deliver. For example, nucleic acids like siRNA or oligos, or for the cell surface, for the cell membrane, for proteins, for specific hydrophobic patches that you want to interact with. So there's a lot of variability that can be built in for these peptides to interact with different types of cargoes. So the strategy that we use using these peptides is to complex these peptides with nucleic acids or proteins or other peptides or peptide nucleic acids, and then condense them into particles and then have them deliver across the cell membrane into the cells. The other important aspect of this technology is that it doesn't rely on endocytosis, and it doesn't have endosomal trapping. So we can get through the cell membrane through a perturbation of the cell membrane and not be entrapped in the endosomal pathway. So this is some data that supports that, that we bypass the endosomal pathway. And here, this EM pictures, and then this is actually looking at calcium influx signals into cells. And this is done with HeLa cells, where you have them in culture, and then you put the particles in culture, and you can measure the calcium influx that happens rapidly as the particle translocates through the membrane. But then the depolarization comes back to normal, meaning that the cell membrane is closed again. Then you can reactivate that with another dose of particles. You get the translocation through the membrane again, and again the membrane closes up. So this occurs without toxicity or minimal toxicity. And you can see these labeled siRNAs that collect inside the cell, and these are not entrapped in the endosome, and these are directly translocated into the cytoplasm. So this is a big difference from sort of the conventional delivery methods where you have to think about endosomal release and all of those aspects. So another piece of data that supports that we can do the transfection in the presence of inhibitors of endosomes. So you can use beta-methyl cyclodextrin, or you could use nocodazole or amiloride, which actually stop the endosomal endosomes from occurring. And in spite of that, you get, this represents the mRNA knockdown for GAPDH, and you get uniform transfection despite those inhibitors. So what can we use this for? Of course, we can deliver siRNA, we can do plasmid DNA, we can do Cas9 gRNA, guide RNA, and for gene editing. We can do it for developing CAR T cells, etc. And what I want to show you today is just a little bit of data on each of these applications, so to familiarize you with the technology. And here we're creating nanoparticles about 120 nanometers in the case of these constructs. Here it's a little bit different. They're smaller because this involves a protein. And our approach that is different in the case of Cas9 is that we're actually delivering the protein and not relying on a plasmid that produces the protein after it's transfected. So this is direct delivery, which probably makes it more efficient. So I'll talk first about siRNA and just go through a series of data. And here you see it's a comparison of the knockdown in different cell types. And these are mouse embryonic fibroblasts and mouse embryonic stem cells. So traditionally, difficult to transfect cell lines, but we can show that clearly in the red bars, this is the what we call the ADGN siRNA complexes, and we compare that to lipofectamine transfection, and looking at the relative knockdown, you can see that dose for dose you get much better knockdown, which is about 8 to 100-fold better than lipofectamine in both of these cell lines. And we have very little toxicity or minimal or no toxicity even at fairly high doses of the peptide up to 100 micromolar. So how is the distribution? This is just one of the peptides that we're using complex to a fluorescently labeled GAPDH siRNA. And you see the interesting thing is that you get a broad distribution, but you don't have the early liver accumulation that you see with typical lipid-based systems. So you get a broad distribution. You do get levels in the liver, but this is after 3 and 5 hours. So it's distributing widely, and then we can do look at the knockdown of GAPDH across all of the tissues in the animal, and you can see that you get quite a bit of knockdown in tissues, even like the brain, where you're getting almost 40% knockdown with a low dose. You have kidney, you have muscle, you have lung, of course you have spleen, you have pancreas knockdown almost 40%. So across a wide variety of tissues, we can get just using this peptide-based platform. It's not limited to liver-based accumulation. We can show this effect in tumor xenografts, and this is a HD29 colon tumor, and we dose these animals. So these are the controls. The blue is the untreated control. We have cyclin B1 is the main target here of the siRNA, and at 2 different doses, at 5 microgram and 10 microgram, and you can see the dose response in the tumor, clearly affecting the tumor size. If you look at on day 44, I believe we can look at the knockdown corresponding to these different groups, and you see clearly that there's quite a bit of tumor knockdown of cyclin B1. And as far as animal weight and toxicity, we don't have really any effects. So you can get the effects directly into the tumor. If we want to combine more than one siRNA to get a dual effect, we can do that. Here we looked at a combination of cyclin B1 and CDC20, both involving the cell cycle. And when you combine the two at sort of half the normal dose, you get a very nice effect on tumor suppression. So we also looked at a factor 7 knockdown, which is a good model to look at. And based on a single injection of the siRNA, at 3 mg per kilo. And this is sort of replicates what has been published with some of the Alnylam methods of delivery. And with a single injection, you get either given IV or subcutaneous. So the blue curve is subcutaneous, the yellow is IV. You get a knockdown of factor 7 that lasts for quite a long time, over a month, and then it slowly climbs back up around day 70. And then we can redose and get the knockdown again. So the point being that you can have subcutaneous injection, you can have IV injection, so depending on the application, and with a single injection you can get a knockdown. We could, of course, increase, give multiple subcutaneous, for example, injections here, and you would get a deeper knockdown. So again, sort of proof of concept, proof of principle, you can do this in different ways and get a deep knockdown. So moving on to plasmids and gene delivery, we can do this in, we get stable transfection using whether it's T cells or, different types of cell lines. And these are plasmids encoding yellow fluorescent protein. What we do is make the constructs with the peptide into the nanoparticles, put them on the cells, and then measure the level of expression of yellow fluorescent protein over time. And you see we get a fairly stable incorporation out to day 14. This is where we stop measuring, but so up to day 14 you can get a good level of YFP expressing cells. Almost it's above 60, above 70%. And you can see all of these different clones which we can sort of clone out and expand if necessary. So expression is maintained over a good period of time. In this case, we tested it for over 14 days. So where do we apply this? We can do, you heard a lot about the CAR T cells and the anti-CD19 CAR T cells. So we've taken that same CAR T vector. In this case, this is a CD19, anti-CD19 vector, second generation as we call it, which has the CD19 specific CFV and then the signaling moieties as well. And we can put this onto T cells, transfect the T cells, and then measure the level of transfection. And you get very good transfection in these T cells. And again, this was measured out to 12 days and is stable expression out to 12 days. And the viability of the cells is quite good. So without toxicity, we can use now a non-viral delivery method to create CAR T cells. And this has obvious advantages for those of you who are familiar with the viral vectors in terms of manufacturing, etc. What we can also do, and recently you might have seen that there was a clinical trial to be initiated with CAR T cells. In addition to the CAR T on the CD19 expression on the CAR T cells, you have PD-1 knockdown or knockout. And this helps to make the T cells more effective because these are checkpoint inhibitors. So what we've done here is first create the CAR T cell, like we just did, I showed you in the previous slide, and then come in with the siRNA in this case for PD-1 and knock down the siRNA, the PD-1. And you can see here that, again, the transfection efficiency is pretty high, and then you can measure the PD-1 levels, and this is just the vector alone does nothing. And then when we have the PD-1 with the peptide, and you can get a very nice knockdown and then the corresponding gap DH was also run, and you see, so as a control siRNA, and then there's no PD-1 knockdown there, of course. So here's a way to do combinatorial therapy and sequential therapy on T cells, create whatever type of constructs that you want to reinfuse into the patient. So the last piece I want to talk about, and I'm running out of time, so just a couple of slides, is application in CRISPR-Cas9. And this is a very hot topic in terms of gene editing. So I won't go into the details. I think you know about it. The concept is you'd use the Cas9 and gRNA, guide RNA to create these edits into genes, or to create or insert nucleotides into genes to make corrections. And we can do all of this. The difference being from the conventional techniques is that we can complex the protein, the Cas9 and the gRNA, the construct together into these nanoparticles and deliver them directly. So we don't have to deliver the plasmid, so the cell then expresses the Cas9, etc. So this is done in a number of cell lines, and these are GFP secreting cell lines, so you can visualize them, U2OS, HEK, K566, a number of them. And then the signal that we look for is if you get the editing, you lose the GFP signal. So clearly you can see here after treatment, you can get a drop, a significant drop in the GFP signal. And if you use what's commercially available, RNAi max, which is a type of lipofectamine, which is in the orange and brown bars, you see very low efficiency. So this is technique using the peptides is at least 7 to 10-fold more efficient than the sort of standard techniques available. Here we've done the same thing, but across, and we'll be looking at actual gene editing of multiple genes at the same time. So the EMX1, AAVS, and HPRT genes, again, across a variety of cell types that include sort of difficult cells like the human embryonic stem cells and neuronal cells, etc. And you can see the level of editing that we get is high in the 80s, 80% range. And if you compare with the RNAi max, you're essentially getting, other than this U2OS cell line, in the human and the embryonic stem cells, you get very little. So this technique is quite efficient. The last couple of slides is the homology-directed repair. So now we're going in, cutting with the CRISPR technique, and then inserting a gene of interest to make the repair. And here, these are just data showing that we can do it all in a single complex. So here we've taken the Cas9, the gRNA, and the DNA that we want to insert and complex them all together into a particle with the ADGN peptide. So you can do it that way, or you can do it where you have just the Cas9 gRNA and the ADGN peptide as one particle, and then the DNA that you want to insert as a separate particle. And here's the comparison of the two. They're fairly comparable, so you could do either or. And then when we compare sort of RNAi max, you get a much lower level of the HDR. So we get significant ability to correct genes using this technique. So to summarize, we have used these short amphipathic peptides that can form complexes with polynucleotides, proteins, and other cargos, and create nanoparticles in the range of roughly about 100 nanometers, effective for a variety of different nucleotides. And we could do CRISPR and gene editing quite efficiently. Important thing is there's a non-endosomal pathway for cell entry, efficient and non-toxic transfection in vitro. And then we have animal model data as well in some cases. So thank you for your attention, and sorry for running over time.
So maybe time for 1 or 2 short questions.
Yeah, very interesting data. I just wonder, the data you showed for the calcium influx and the experiments with the inhibitors of endocytosis, was that just for the peptide, or even when it was bound to particles of 120 nanometers?
No, this was for the complex. So for the siRNA and peptide complex. Because we showed the knockdown doesn't change with GAPDH. It doesn't change if you have those inhibitors in the system as well.
And was that particles of 120 nanometers even?
Yes, yes.
Okay.
Yeah, about 100 nanometers, 120, yes.
So do you see any immune response? Can you retreat animals several times? Or?
Yeah, so we've done that. In the tumor models, we retreat them all the time. So every few days, we can give an injection. So there's no issue. We've also, I didn't show the data, but we've run these cytokine panel kits that you get, like a 20-plex cytokine, and it's very low, if at all, levels of stimulation. So we think that the peptide is masking the oligos very well, and so the body doesn't really see it.
Maybe one last question up there. Could you wait till the microphone is on?
Yeah.
Hi, thank you. I was just saying this is a very nice, clear presentation. With your in vivo distribution, did you see any evidence of it going into the PBMCs?
We didn't look for that. But,
would you expect it to be there?
Yeah, I think we do. We do. And because our transfection, at least in vitro, the transfection works very well with the T cells and other blood cells. So we haven't looked and measured in vivo, but that's clearly something we can look at.
So, thanks,
Okay, thank you very much.
Once again.